In this explainer, we're going to break down a recent study that demonstrates how specific chemical compounds can reverse key markers of cellular aging. The research explores a non-genetic method for rejuvenation, based on the idea that aging is driven by a loss of epigenetic information.
So here's our roadmap. We'll start with the theory of aging this research is built on, then we'll look at the scientific method they used, what they discovered, and what it might mean for the future of regenerative medicine.
First up, an information theory of aging. To really understand how you might reverse aging, you first have to understand how our biological information is stored, and more importantly, how it can degrade over time.
Think of your cells like a computer. The genome is the hardware. It's stable, digital information, the fundamental genetic code that's consistent across your cells. But then there's the epigenome, which is more like the software. It's an analog layer of information that tells the hardware what to do, which genes to turn on or off. And like old software, this information can get corrupted over time.
And that brings us to the core of this theory. It proposes that aging isn't fundamentally about the genetic hardware breaking down. It's about the epigenetic software getting corrupted. The cell effectively loses its ability to properly read its own instruction manual. This loss of information is thought to be what drives the cellular decline we recognize as aging.
So if the problem is corrupted software, the next question is, can you restore it from a backup? Well, the concept of resetting this epigenetic information isn't new. It was actually established first using genetic methods, which set the stage for this study's chemical approach.
A landmark discovery showed that expressing a few specific genes, known as Yamanaka-factors, could effectively rewind the clock on cellular age in animal models. It's thought that they access and restore a sort of backup copy of youthful epigenetic information. This was the proof of concept. The challenge, however, is that genetic methods are expensive and carry significant safety concerns, which is why the search began for a chemical alternative.
Okay, so if you want to find a chemical solution to reverse aging, you first need a tool. You need a way to observe and measure cellular age quickly and reliably, especially if you're going to test a large number of compounds.
And this was the central problem to solve. You have to develop a rapid, scalable assay, a test that can reliably tell young cells from old ones. This is the only way to sift through countless possibilities to find something that actually works.
The researchers decided to focus on a well-known hallmark of aging, the breakdown of the boundary that separates the cell's nucleus from its surrounding cytoplasm. In young cells, this separation is distinct. In old cells, it becomes leaky. This is called a loss of nucleocytoplasmic compartmentalization, or NCC.
So they engineered a clever visual system to track this. In their model, a green fluorescent protein is kept out of the nucleus, while a red one is kept in. In a young cell, the green and red stay separate, but in an old, senescent cell, that boundary deteriorates and the proteins start to mix. This mixing of colors provides a clear, real-time signal that a cell is in an aged state.
Armed with this NCC assay, the researchers screened 80 different chemical combinations. They applied them to senescent human cells to see if any of the cocktails could restore that youthful boundary and, well, unmix the colors.
The screening process was successful. Out of the 80 combinations they tested, 6 distinct chemical cocktails were found to reverse the signs of aging in the NCC assay. These were all based on one of 2 core formulas, with different additives known to influence cellular processes like metabolism and growth.
Seeing the colors separate was a promising start, but the next step was to go deeper and quantify the effect. Did these chemical cocktails actually make the cells younger on a molecular level? To find out, they needed to look at the cell's gene expression profiles. This brings us to the concept of transcriptomic age. It's a way of measuring a cell's biological age by analyzing its patterns of gene expression, which genes are currently turned on or off. These patterns change predictably with age, so they can be measured by what are known as computational clocks.
The transcriptomic analysis confirmed what the visual assay suggested. All 6 of the chemical cocktails produce a statistically significant reduction in the transcriptomic age of the treated cells compared to the untreated old cells.
So, how much of a reduction are we talking about? When measured with a clock specific to human cells, the most effective cocktails wound back the cell's estimated age by more than 3 years. This occurred after only 4 days of treatment. Now, here is a critical finding from the study. This rejuvenation happened without completely erasing the cell's identity.
The treatments did not reset the cells all the way back to a pluripotent or stem cell-like state. This is an important safety consideration because inducing full pluripotency carries the risk of uncontrolled cell growth. This result suggests rejuvenation can potentially be separated from that risk.
These findings mark a notable step in the development of rejuvenation medicine. They demonstrate that some reversal of aging markers may be achievable through chemical means, not just genetic ones. So what are the implications? To summarize, this study shows that both genetic factors and small molecule chemical cocktails can successfully reverse key biomarkers of cellular aging. In both cases, they were able to take a senescent cell and restore a more youthful gene expression profile.
So the key finding is that these chemical cocktails can reverse transcriptomic age in human cells. The mechanism appears to involve turning up cellular energy pathways while turning down inflammatory pathways. This offers a potential non-genetic route for regenerative medicine. Future work will need to focus on the precise mechanisms involved and, of course, extensive safety testing in animal models.
This study suggests that our old cells may not have forgotten how to be young, but have simply lost the correct instructions. And this leaves us with a fundamental question. If aging is indeed a loss of information, how much of that information can truly be restored? And what would that ultimately mean for the future of human health?